My dear students, welcome to my today's online class on the open elective paper, that is Parasitology. In the last class, we had discussed questions from 43 to 55 and got familiar with the various parasites, their taxonomic positions and their binomial nomenclature, etc. And now we are left with few more questions to discuss, and the question number 56 goes like this. It has two parts, A and B. Students, sometimes like this also questions can be asked for 2 marks. The 2 mark can be split into 2 halves, each carrying 1 mark, and it can be like A, B. Now as we have here. Better get ready for these type of questions also. And very easy to answer also. And now this question number 56A goes like this. ELISA is used for the diagnosis of AIDS. We have discussed in our previous classes about various molecular diagnostic techniques, and one such technique happens to be ELISA. To remind you, it stands for enzyme linked immunosorbent assay, and in that context we had discussed that it is used as a diagnostic tool for viral infections, and especially in the diagnosis of AIDS. Okay, so it is not diagnostics, it is diagnosis. Okay, ELISA is used in the diagnosis of AIDS. You should tell whether the statement is true or false, and the answer is true because it is used for the diagnosis of AIDS, acquired immunodeficiency syndrome. 56B, the question goes like this. Vampire bats spread a disease called rabies. Rabies andre kelidiralla, huchunayi kadutta anta. Again, it's a viral infection. And you must have heard of vampire bats. We discussed that they are swangivorous mammalian ectoparasite, the only mammal which feeds on the blood of other animals, right? So the question is, vampire bats spread a disease called rabies. True or false? For your convenience, we have made the correct answer bold. We have put the correct answer bold. So here also the statement is true. If you just write T or UE, you will get 1 marks. Okay, or if it is false, if you write false, you will get 1 mark. So be ready to answer these type of questions also. Then the next question is also of same type. 57A. Hood marking bird, that is mimus, is endemic to Galapagos Islands. You must have watched in your this one Zoo Geographic channel and all. They would have shown the fauna of these Galapagos Islands, and here you should know the term endemic. What is endemic? If a particular animal or a plant species is restricted to one particular region on this globe, then we say that particular species is endemic to that region. So here the question is, food marking bird is endemic to the Galapagos Islands. That means it is not found anywhere else other than in Galapagos Islands. And the answer is very much true. It is endemic to Galapagos Islands. Like this, we have a few species which are endemic to our Western Ghats, like that. Okay. Then 57B is CFT, that is complement fixation test, another diagnostic tool, molecular diagnostic tool for infections. CFT mainly measures IgG antibodies. It is true because CFT basically works with that. IgG antibodies are one category, one type of antibodies which are produced as a result of infection in the body. Answer is true again. All the time answers need not be true as I have given here. It can be false also. Okay. Question will be just changed like CFT mainly measures IgM antibodies. Then the answer is false. That's all. Okay. Then next question is also of same type. 58A, CCI, that is countercurrent immunoelectrophoresis, is used for detecting pneumococcal capsular antigens. We have studied this and the answer is true. And 58B, it should have been B, sorry for the typographical error, B. coli, that is Blantidium coli, inhabits frog's rectum. Here is one small mistake, I want to correct it. Answer has been given false. No. B. coli is basically a parasite living in the rectum of, inhabiting the rectum of many animals like monkeys, pigs, etc. So by eating pig flesh, that's how the humans contract this infection of B. coli. But it do inhabits the rectum of frog. Answer is true only. There should have been a slight change in the question. The question should have been like this. B. coli, Blantidium coli, inhabits frog's rectum also apart from other hosts like monkeys, pigs, etc. If that were to be the question, answer is true. Okay. So B. coli do inhabits frog's rectum. In earlier days when we were doing dissections, after dissecting the frog for its arterial system or pectoral or pelvic girdle or so, finally we would ask the children to cut the rectum part, cut open, take the contents on a watch glass and observe under the microscope and they would see the Blantidium coli. Okay. That is a ciliate protozoan. So the answer is true. Only thing is question should have been like this. B. coli inhibits frog's rectum also apart from other hosts. Okay. That's all. Then dear students, questions can be in the form of match the following. The 59th question is like that. In the column A, okay, we have given various molecular diagnostic tests or techniques. In the B, we have given certain matches and you need to match. For your convenience, in the column B, we have given in the brackets the number also that telling you the correct answer. For example, CCI, countercurrent immunophoresis, is the immunoelectrophoresis technique. You know that it is for pneumococcal antigens. So, in the bracket we have given answer as 1, right? Then ELISA, you know that it is used for diagnosis of AIDS. CFT is for detection of IgG antibodies and PCR, polymerase chain reaction, is for amplification of DNA fragments. So, in the bracket we have given the answers also, right? All this has been explained while discussing the questions on molecular diagnostic tools. Okay. So, for each correct match you will get half marks. You can expect questions like this also. Alright. Now, here is a question like descriptive question. What are DNA probes? Mention its uses. In the previous sessions, we have learned what are molecular probes, right? And molecular probes can be of two types, DNA probes and RNA probes. So, DNA probe and its uses, okay? DNA probes are single-stranded sequences of DNA. By now, I assume that you know what is DNA. DNA is deoxyribonucleic acid. RNA is ribose nucleic acid. These are two genomic materials which are found in animals and plants, microbes, etc. So DNA probes are single-stranded sequences. They are also known as gene probes of DNA or RNA. They are basically used to search for its complementary sequence in a sample genome. You will have a sample genome and you will introduce these probes. Mostly these probes are radioactively labeled probes, and they go and bind, hybridize with the complementary sequence on the sample genome. And where all they go and bind, since they are radioactively labeled, when you do autoradiography, you will see the radioactivity on that photographic plate in that particular region. This is how DNA probes work. And these researchers, use DNA probes to screen the genome, looking for the identification of cancers or hereditary syndromes. So in medical field, basically they are used for identification of or diagnosis of cancers or tumors or even hereditary syndromes. These probes are also used to identify microorganisms. These probes find use in Southern blotting technique. One more technique in biotechnology what we use, that is nucleic acid hybridization technique. And they are also used in DNA profiling, DNA fingerprinting and karitivilla, adral kuda balastive. So these are all the applications of DNA probes. The way you have DNA probes, you have RNA probes also, and that is question number 61. RNA probes are stretches of single-stranded RNA molecules. They are used to detect the presence of complementary nucleic acid sequences or target sequences by hybridization. They are usually labeled, for example, either with radioisotopes or epitopes or biotin molecules or fluorophores to enable their detection. So this is about questions on DNA probes and RNA probes. And sometimes the question can be like this: what are the differences between DNA probes and RNA probes? These are all short answer questions, again asked for 2 marks only. The differentiating factor between the DNA and RNA probes is that DNA probes are DNA fragments complementary to that of target sequences of nucleotides. On the contrary, the RNA probes are extensions of single-stranded RNA complementary to that of sequences of nucleic acids of the target sequence. So, dear students, these are all few questions what I wanted to discuss today. There are 62 questions given in the question bank, each carrying 2 marks. But other than that, I have framed 4 more questions which are not there in the question bank, but I want you to listen these also. If you keep a piece of paper and pen with you and take down this, as they are not found here, I am going to discuss. There can be a question like, what are miticides? ನೀವು ಮೈಟ್ಸ್ ಬಗ್ಗೆ ಓದಿದಿರಲ್ಲ, ಟಿಕ್ಸ್ ಆ್ಯಂಡ್ ಮೈಟ್ಸ್ ಬಗ್ಗೆ, ಕೋರೆ ಏನು ಅಂತ ಹೇಳ್ತಾರೆ ಅಂತ. I have discussed about this mites. So in that context you should know what are miticides. You must have heard of herbicides, pesticides, like that. These miticides are the chemicals used to control mites or ticks. ಉಣ್ಣೆ ಮತ್ತು ಕೋರೆ ಎರಡರ ಕಂಟ್ರೋಲಿಗೆ ಕೂಡ ಇದನ್ನು ಬಳಸ್ತಾರೆ. Especially for those species of ticks and mites that damage ornamental plants or food plants. So agriculturally important plants, ornamental plants control where there is heavy damage by these ticks and mites. For the control of these ticks and mite species, people use miticides. ಅದನ್ನ miticides ಅಂತ ಕರೀತಾರೆ. They are all chemicals. For example, azobenzene. Azobenzene is one such miticide. Dicophol is another miticide. Ovex, O-V-E-X, Ovex. Then tetradiflan, another miticide. So these are all the chemicals or the miticides which are used to control mites or ticks, especially those species of mites or ticks that damage the ornamental or food plants. So question number 63 is what are miticides, and answer I have given. Question number 4 is what is pediculosis captitis? You know that pediculosis ಅಂದರೆ ಏನು, ಹೆಡ್ ಲವ್ಸ್ ಅಂತ ಕರಿತೀವಲ್ಲ, ಅದು ಪೆಡಿಕ್ಯುಲಸ್. ಸೊ ವಾಟ್ ಈಸ್ ದಿಸ್ ಪೆಡಿಕ್ಯುಲೋಸಿಸ್ ಕ್ಯಾಪ್ಟೈಟಿಸ್? ಈ ಚಿಕ್ಕ ಮಕ್ಕಳಲ್ಲಿ ಕ್ಲೀನಾಗಿ ಇಟ್ಕೊಳ್ದೇ ಇದ್ದರೆ, ತಲೆ ಎಲ್ಲ ನೀಟಾಗಿ ತಲೆ ಸ್ನಾನ ಮಾಡ್ಕೊಳ್ದೇ ಇದ್ದರೆ ಈ ಹೇನು ಚೆನ್ನಾಗಿ ಇನ್ಫೆಸ್ಟ್ ಆಗಿಬಿಡುತ್ತೆ ಆನ್ ದ ಸ್ಕಾಲ್ಪ್, ತಲೆ ಬುರುಡೆ ಮೇಲೆ. ದಟ್ ಕಂಡೀಷನ್ ಈಸ್ ಕಾಲ್ಡ್ ಪೆಡಿಕ್ಯುಲೋಸಿಸ್ ಕ್ಯಾಪ್ಟೈಟಿಸ್. ಸೊ ಪೆಡಿಕ್ಯುಲೋಸಿಸ್ ಕ್ಯಾಪ್ಟೈಟಿಸ್ ಈಸ್ ಅ pathological condition ಅಂತ ಹೇಳಲಿಕ್ಕಾಗಲ್ಲ, is a condition in which the tiny insects infest the scalp ಮೇಲೆಲ್ಲ ಕೂತ್ಕೊಂಡ್ಬಿಟ್ಟಿರುತ್ತೆ. ಆ್ಯಂಡ್ ಎಕ್ಸಾಕ್ಟಾಗಿ ರೋಗ ಅಂತೇನು ಬರಲ್ಲ, ತಲೆ ಎಲ್ಲ ಕೆರೆಯೋದು, ಆ್ಯಂಡ್ ದೆನ್ ಆಲ್ ದ ಟೈಮ್ ಹ್ಯಾವಿಂಗ್ ದ ಸೆನ್ಸೇಷನ್ ದಟ್ ಸಮಥಿಂಗ್ ಈಸ್ ಮೂವಿಂಗ್ ಆನ್ ಯುವರ್ ಹೆಡ್, ಏನೋ ಓಡಾಡ್ತಾ ಇದೆ ತಲೆ ಮೇಲೆ ಕೂದಲೊಳಗೆ ಅಂತ ಅನಿಸೋದು. ದೀಸ್ ಆರ್ ಆಲ್ ದ ಸಿಂಪ್ಟಮ್ಸ್. ಆ್ಯಂಡ್ ದಟ್ ಕಂಡೀಷನ್ where these tiny insects, normally they are head louse. There are few other species also, but majorly it is the head louse or human louse, which infest the scalp. So pediculosis captitis ge uttara enu? Pediculosis captitis is a condition in which tiny insects infest the scalp. Okay, right. Then there is one more question. Question number 65. Again, question I have divided this question into two parts. I will give only one question here. Fleas carry the organisms of bubonic plague or murine typhus. Fleas andre chigita anta karitaralla. We have studied this in our earlier session. ಸೊ ದೀಸ್ ಫ್ಲೀಯಾಸ್ ಕ್ಯಾರಿ ದಿ ಆರ್ಗನಿಸಮ್ಸ್ ಆಫ್ ಬ್ಯುಬೋನಿಕ್ ಪ್ಲೇಗ್ ಜ್ವರ ಅಂತ ಮುಂಚೆ ಬರೋದು, ಇಲಿಗಳ ಮೂಲಕ ಹರಡೋದು, ಶರೀರದಲ್ಲಿ ಗಂಟು ಕಳಿಸ್ಕೊಳ್ಳೋದು. ಇಟ್ ವಾಸ್ ಟ್ರೀಟೆಡ್ ಆಸ್ ವೆರಿ ಎಂಟಮಿಕ್. ವಿಲೇಜಸ್ಸೇ ಎಲ್ಲ ಸತ್ತೋಗ್ಬಿಡೋರು ಜನ. ದಟ್ ಬ್ಯುಬೋನಿಕ್ ಪ್ಲೇಗ್ ಆ್ಯಂಡ್ ಮ್ಯೂರೈನ್ ಟೀಫಸ್. ಸೊ ಆನ್ಸರ್ ಈಸ್, ಇದು ಟ್ರೂ ಆರ್ ಫಾಲ್ಸ್ ಅಂತ ಕ್ವಶನ್ನು. ಆನ್ಸರ್ ಈಸ್ ಟ್ರೂ, ಬಿಕಾಸ್ ಫ್ಲಿಯಾಸ್ ಆರ್ ದ ಒನ್ಸ್ ವಿಚ್ ಕ್ಯಾರಿ ದಿ bubonic plague and murine typhus. And 65B, it can be any question like this. Cookie cutter shark is Istius. ಮುಂಚೆ ಹೇಳಿದ್ದೀವಲ್ಲ, cookie cutter shark is a mesopelagic species. True or false? And the answer is true. So this is how 65A and B can be given. So questions can be asked this way also. 65A is about fleas, 65B is about cookie cutter shark. Okay. Students, I have one more questions. Last question for you people, and I'll end my this talk with that answering that question. 66th question. Please take down. What are mesopelagic animals? Give an example. What are mesopelagic animals? Give an example. Mesopelagic zone, also known as middle pelagic or twilight zone, is part of the pelagic zone that lies between photic epipelagic and aphotic bathypelagic zones. So, ಈ pelagic zone ಅಂತ ಒಂದಿದೆ. ಮೇಲ್ಗಡೆ ಭಾಗ ಎಲ್ಲಿ ಲೈಟು ಎಂಟರ್ ಆಗುತ್ತೆ, ಸನ್ಲೈಟು, ದ್ಯಾಟ್ ಈಸ್ ಫೋಟಿಕ್ ಎಪಿಪೆಲಾಜಿಕ್ ಝೋನ್ ಅಂತಾರೆ. ಎಲ್ಲಿ ಲೈಟು ಎಂಟರ್ ಆಗಲ್ಲ, ಡಾರ್ಕ್ನೆಸ್ ಡೀಪ್ ಇನ್ ದ ಸಿ, ಅದು ಇಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಎಫೋಟಿಕ್ ಬೇಥಿಪೆಲಾಜಿಕ್. ಓಕೆ, ಎ ಅಂದರೆ ಇಲ್ಲ ಅಂತ, ಫೋಟಿಕ್ ಅಂದರೆ ಲೈಟ್, ವೇರ್ ದರ್ ಇಸ್ ಟೋಟಲ್ ಡಾರ್ಕ್ನೆಸ್. ಬಟ್ ದಿಸ್ ಮೆಸೋಪೆಲಾಜಿಕ್ ಝೋನ್ ಈಸ್ ಸಮಥಿಂಗ್ ಇನ್ ಬಿಟ್ವೀನ್ the photic epipelagic zone and aphotic bathypelagic zone. Okay, and this is a twilight zone ಅಂದರೆ ಡಿಮ್ ಝೋನ್ ಮಿಡ್ಲ್ ಝೋನ್ ಅಂತ ಕರಿತೀವಿ. ಜಾಸ್ತಿ ಬೆಳಕು ಇರೋದಿಲ್ಲ, ಪೂರ್ತಿ ಡಾರ್ಕ್ನೆಸ್ ಕೂಡ ಇರೋದಿಲ್ಲ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಮಿಸೋಪೆಲಾಜಿಕ್ ಝೋನ್. And species, animal species living, inhabiting this area are called mesopelagic animals. One best example for this is your cookie cutter shark. Okay, my dear students, with this I have discussed some more questions what were left undiscussed, and we have had quite a few sessions of online classes. And next Tuesday you can join me for an offline class where I am going to discuss all these videos, because I want to discuss them offline also. Okay, thank you for your patient listening. Stay blessed. See you on Tuesday. Okay.